Hello everyone. In this presentation I'm going to start a series of lectures that are about cell signaling. In this first lecture I'm going to talk about signaling in general, about the various receptors that are involved, particularly and specifically I will detail intercellular or nuclear receptors and G protein coupled receptors. First of all, we have to clarify what signaling is. This figure is rather busy and it's intended to convey to you that the studies related to cell signaling are not very easy. They are not meant to be. They are rather complex. And when you look at this figure, the first thing that strikes you is that there's lots of names on it. So you will be running into a problem that you will have to remember a lot of 3 and 4 letter words, and 4 letter words are not always beautiful, as we know. And then a lot of interactions between these entities abbreviated by a few letters. Obviously this is not a scheme that you will need to memorize. It only illustrates the difficulty that you face, and we certainly will not be going into such complexity. Then the next thing that I have to say is that what this figure would like to symbolize is a general concept of cells receiving messages like trying to find the pointer. Okay, there we go. So cells receiving messages in the form of messenger molecules most often, like in this figure it's platelet-derived growth factor or PDGF, and they express receptors for these messages, and then these receptors serve to interpret the message, get it translated through some complicated system inside the cell, and eventually provide a response or an output. As mentioned before, signals can not only be molecules like the PDGF in the previous figure, but can also be electrical signals that you have partially discussed when you talked about ion channels, and there will be a little more discussion on this when we summarize some basic knowledge on signaling in the nervous system a couple of lectures ahead from now. As discussed in the previous figure, in general when we talk about signaling, it's a process which we can conceive as starting with encoding and emission of a message by one cell, like the cell that secretes the platelet-derived growth factor in the previous example, which is the source cell. And then this message reaches the target cell somehow, and this target cell receives it, interprets it, decodes it, and gives a response. However, for practical purposes, simply because it's so much easier to take a cell in a petri dish in vitro in an experimental system and sprinkle some of the substances that can serve as messengers on it, and then observe what the cell does. We discuss in most cases this process starting from the middle, starting with the molecular details of receiving and interpreting the signal and giving a response, which of course in many cases could be the secretion of another signal. The next issues to be considered are the length of the pathway and the mode of transport of the signal molecules between the signaling cell and the target cells. The most known pathway that you probably are most familiar with is the endocrine pathway. It basically means that a gland in the body, for example, the beta cells of the pancreas, uh, secrete a hormone or a messenger, like the beta cells secrete insulin, as you have already discussed this previously. And this secreted molecule gets into the bloodstream, and in the bloodstream it gets to all the distant target cells in the body, so the path lengths can be up to several meters, but certainly from any part of the body to any other part. Matrix, or you can also call it the interstitium, the material between cells, is the medium through which the message molecules commute from the secreting cell to the neighboring target cells. The neighboring cell doesn't really have to be exactly across the secreting cell. Para, as you remember from your chemistry studies, means across something, but the target cell can of course be anywhere around in a few microns or in maybe a millimeter or 2 millimeter environment of the secreting cell. So, the secreting cell releases the message molecule, it diffuses through the interstitium, and then it binds to receptors on or inside perhaps, but mostly on the membrane of the target cells. This mode of signaling has various subcases. One subcase that we mentioned first is the autocrine case. Autocrine implies that the secreted ligand can actually be bound by the same cell that secretes the ligand, because this cell expresses the receptors for the ligand. In a most special case, which you call intracrine, the ligand is not even secreted, but it's bound to receptors that are also still located intracellularly in this signaling cell. When you consider the concept of secreting a signal and then binding it by the same cell, at first sight it resembles a reasonably pathological condition of when somebody talks to himself or herself. So this is usually not good, and in the cellular concept, sometimes it's also not good. It's not good in the case of tumors, because tumors can secrete growth factors that stimulates their own growth. So a tumor cell can secrete a growth factor like PDGF that we had in our first example, and express the receptor for it, and then will stimulate its own division, which is coherent with the tumor's nature of uncontrolled cell division. This kind of autocrine signaling is not purely autocrine, but it can and usually is joined by paracrine signaling, because the secreted ligands also stimulate all the neighboring tumor cells, and all the neighboring tumor cells can secrete the ligand to stimulate the others. So it's a mutual enhancement situation, very much like to the situation of a football game where all the audience suddenly stands up and shouts nice or not so nice words to the players when they are enthusiastic, or just the opposite. Of course, this is not the only case where we have autoparacrine signaling, because it has its own place in regular normal biological signaling processes. For example, during embryonic development, a group of cells can secrete a substance which is used to stimulate coherence between or among these cells, to drive them to develop into a certain given common direction. For example, a little extremity bod which becomes a little foot or a little arm later on. Okay, so on the next slide we will look at more subtypes. Let us now consider the other subtypes of paracrine signaling. The first one that you see here on the 2B is directed secretion. The major difference is that you have some molecules in the membrane of both the signaling cell and the target cell which bind to each other, so they keep the two cells together for the duration of signaling, which means that the secreted message mostly reaches the target cell and only a little bit is leaked to the neighborhood. A specific example would be killing of target cells by cytotoxic lymphocytes, or the activation of B lymphocytes by helper T lymphocytes, which we will study in more depth in a later lecture on signaling. A special subcase of directed signaling or directed secretion would be synaptic signaling, which is characteristic of the nervous system. And at this point I just want to emphasize the very structured nature of the contact between the two cells, the presynaptic and the postsynaptic cell, which together form the synapse. And in anatomy and histology you will learn a lot about the connecting structures between the two cells, which are far more complex than a simple ligand receptor interaction that you had in directed secretion. Also, for this signaling, vesicular transport is very characteristic, which you have partially in quite a bit of detail studied previously in this course, and when we talk about the nervous system we will return to it. Finally, as the last subcase, I wish to mention the case of the membrane-bound ligand. This is also called juxtacrine or contact-dependent signaling. It's called contact-dependent because the signaling cell and the target cell need to be in each other's contact, neighboring each other, because the signaling cell expresses the ligand as a membrane-bound molecule, and the receptor on the target cell of course is membrane bound, so the two cells should be touching each other for the signaling to occur. An example that you will be studying in more detail is the interaction between delta and notch in the development of the nervous system, but later on in your studies I'm quite sure that you will be meeting the Fas ligand system, which is a pathway stimulating cell death. The last big group of signaling pathways is the pathway that relates to the gap junction. Gap junctions you have learned about. They are channels connecting two cells by the coupling of two connexons, one in the membrane of each cell, and these connexons are made of connexin subunits, forming a channel which is conductive to molecules up to 1 to 2 kilodaltons, regardless of charge and size, up to of course 1 to 2 kilodaltons. So one of the interesting possibilities, in addition to passing larger molecules through gap junctions, is that they can conduct ions, and therefore in the case of an electric synapse in the nervous system, the depolarization wave, when it arrives to the presynaptic cell and causes an ion disbalance and ion movement on that side. This electric charge and the depolarization it represents can easily pass through the gap junction, and it's a very fast way of transferring the depolarization to the postsynaptic cell, which can then forward it. In this particular example, you see a cell culture which is adherent to the bottom of a dish. It's a glioblastoma cell culture in monolayer, and among the cells you have gap junctions. So if you scrape the cells with a pipette, you hurt the cell membrane around the reasonably intact cells around the scrape. And if there's a fluorescent dye, like this guy Lucifer yellow in the petri dish, then these cells will take up the dye, and through gap junctions it will be passed on to neighboring cells. I want to remind you that gap junctions are regulated by calcium concentration, which high calcium concentration closes them. Also phosphorylation is a regulatory path, and these regulations are important physiologically. There are also other ways of regulating them. For example, mechanical contact or mechanical stimulus will also decouple the gap junctions, so they will separate from the connections will separate from each other. As you probably realized, there are two types of people: one who like to classify, and the others who don't like to classify. Well, I'm in the first category, so I'm a friend of classification. You will experience more of this later on. I believe it's useful because it helps you remember things like how many items you need to list to get a more or less complete coverage, or it helps you understand the relation or interdependence of various concepts. In this particular case, when you classify ligand receptor interactions, then the easiest thing is to go by the solubility of the ligand and say, okay, the ligand can be either hydrophilic or hydrophobic. For a hydrophobic ligand, it's easy to pass through the membrane of the target cell, which implies that it can have both intercellular receptors, because the ligand can go through the membrane and bind to the receptor inside the cell, but it can also have a receptor in the cell membrane, so that is a rather rare case, but it can happen. The other possibility that the ligand is hydrophilic precludes the probability that the ligand can go through the cell membrane, and implies that the receptor for the ligand must obligatorily be located in the cell membrane of the target cell, with the ligand binding domain protruding to the extracellular space. So the ligand that is not able to penetrate the membrane can also be bound, and is signal forwarded into the inside of the cell. So in our discussion of various receptors and their modes of working, we will start with the smallest possible group, so that we can do it in the least painful manner possibly, which is the group of intercellular receptors, which are the receptors for most of the hydrophobic ligands. Of course, you can also say lipophilic, which is roughly the same. These ligands mostly fall in categories like steroid hormones, thyroxine, or vitamin D3, and each of these categories probably rings a bell. You can have an example of Arnold Schwarzenegger, who is probably not on steroids but could as well be. and of course you have all the other sex hormones which fall in the steroid category, but also mineralocorticoids and glucocorticoids that you have already studied in high school. And one other item that's worth mentioning is thyroxine. Thyroxine is related to the thyroid gland, and a disease related to it is goiter, and then the last group is the last item that I want to mention is vitamin D3, which is also related to a very well-known disease, rickets, which tells you that vitamin D3 has one major role of depositing calcium in bones and preventing bones from bending like they do in the absence of the vitamin on this picture. The common characteristic of these hormones that bind to such intercellular receptors are their direct effect on gene transcription, because their receptors are in fact transcription factors. And through gene transcription they produce new proteins that affect cell cycle, metabolism, and related issues. Because these hormones binding to intercellular receptors are lipophilic or hydrophobic, then they're not too happy in the bloodstream alone, so they tend to bind to carrier proteins that shade their hydrophobic surfaces from the hydrophilic blood or liquids in the blood. They diffuse through the capillary and through the interstitium they get to their target cells. They are, I would say, at least for these examples, all or endocrine type hormones that travel all around in the blood. And once they get to their target cells, wherever in the body, they diffuse through the cell membrane because they are lipophilic and bind to their receptors, and the receptors bind to the DNA and promote transcription. So, as I said, they are transcription factors. One major, I'm sorry, so one major subtype of these receptors works in a way that the receptor resides in the cytosol. It is bound to an inhibitor, which can be a heat shock protein or an immunophilin, and when the hormone diffuses into the cell, binds to the receptor, then the receptor changes conformation after binding the hormone with its hormone binding domain, releases the inhibitor, can diffuse, get through the nuclear pore, get into the nucleus, where in dimer form it binds to DNA. I would like to emphasize that it is not important whether it gets through the nuclear pore in dimer form or not. There's dimer monomer equilibrium both in the cytosol and in the nucleus, so this is not an important issue. You should even forget that I mentioned it. The thing, however, that you should remember is that the hormone binding exposes the DNA binding domain of the receptor, so it's happily binding to DNA, and the stretch of DNA it binds to is called the hormone response element, because that should orchestrate the response to this particular hormone arriving to the cell. And once the activated receptor binds to the hormone response element, it will recruit an activating complex which promotes transcription of the regulated gene that this hormone response element is regulating. In this table, you see again a classification, and that divides these intercellular nuclear receptors into two categories: homodimer receptors and heterodimer receptors. What we have discussed in the previous figure is characteristic for homodimer receptors, and these cover five, so that should be easy to remember. They all are steroid structures: estrogen, progesterone, androgen receptors, and then the glucocorticoid and mineralocorticoid receptor. So, so the fact that they are homodimeric means that when this dimer is formed that I showed in the previous slide, this is a dimer between two identical receptors, like two estrogen receptors binding both estrogens, or two progesterone receptors binding each progesterone molecule, and so on and so forth. And and as we said, they are inactive in the cytosol, bound to some co-repressor or inhibitor, and once they are in the nucleus, bound to the DNA after they bound the ligand, the activating complex that they recruit has a histone acetylase activity, which, as you learn from molecular biology and probably also genetics, histone acetylation will cause the increase of negative charge on histones, which repel the negative charge of phosphates in DNA. So this process, this histone acetylation, then eventually causes the loosening of the DNA on the nucleosome, and a better accessibility to RNA polymerase and the start of transcription. The other category of intercellular receptors belong to the homodimeric group, and there's not many that we have to remember here. We have mentioned vitamin D3 and the thyroid hormone, and the receptors for these belong to this group, and also for the receptor for retinoic acid. The fourth group that belongs here is the group of orphan receptors. They are called orphan because the receptors were known, but there were initially no known ligands. But of course, no receptor remains orphan. All of them have a purpose, and then consecutively, the ligands for most of these orphan receptors have been discovered. For example, there are various lipid receptors that regulate metabolism. The characteristic of these receptors, the reason why they are heterodimeric, is because each of these, the retinoic acid receptor, the vitamin D receptor, the thyroid receptor, they all pair up with somebody else and not an identical receptor. And this somebody else is in most of the cases called the retinoid X receptor, or RXR, which can form dimers with each of these hormone receptors to complement and generate an active structure. The difference is not only in being heterodimeric with RXR when compared to the homodimeric receptors, but also that most of these receptors are already in the nucleus also when they are inactive in the non-ligand bound form. So they form heterodimers in various equilibria with monomers, and the heterodimers can bind to the DNA, but without the ligand they bind a so-called co-repressor, which helps maintain inactivity. And the moment the ligand arrives and gets through the cell membrane and gets into the nucleus, binds to the specific receptor like RAR or VDR or the thyroid receptor, then the co-repressor is thrown away, and a co-activator complex is recruited, which again has the major activity of histone acetylation, promoting the loosening of nucleosomal structure and the start of RNA transcription. So as you saw, this was a very, very small group of receptors and only 8 names to remember and 2 categories. And unfortunately, we can't be very happy with this because we have to turn to the next big group of receptors, which are located in the cell membrane, and this is a huge group. We have discussed that obligatorily this group of receptors must be serving for binding all the hydrophilic ligands, like as example we can say epinephrine or peptide hormones, but also some hydrophobic ligands can be bound by cell membrane receptors, and here we mention the example of prostaglandins. Obviously, because these receptors are in the cell membrane, they serve to translate the message which arrives in the form of a ligand, binds to them, and translation very often happens through second messengers. These second messengers, like these little green balls here, exist already in the cell, but maybe not at very large numbers. And when the message arrives and the receptor binds the messenger molecule, then their amount increases, and that forms a signal to the inside of the cell. This means that after the receptor binds the ligand, there are already immediate changes in the molecular constitution of the cell, which can be translated into some very direct and fast responses given by the cell, including cell motion or secretion of some substances, or putting out transporters or receptors into the cell membrane and stuff like this. However, these receptors also have the possibility to also regulate gene transcription like the nuclear receptors do. But since they are in the membrane, of course they have no access to DNA directly, so they must work indirectly through various cascade mechanisms. When the activated receptors produce something, and this something does something, and that activates something, and so on and so forth, all the way down until you get a transcription factor binding to DNA. We will go through various pathways that actually lead to the nucleus and discuss the various mechanisms, mostly in the next and the following lectures. So here comes classification again. I have put cell surface receptors into 3 basic categories, and these categories constitute those receptors that have no enzyme activity, or not their own enzyme activity. It's also called intrinsic enzyme activity, means that it is inside, inherent to the receptor itself, and this group doesn't have that kind of enzyme activity. Obviously, the opposite would be that all those receptors that do have their own enzyme activity, and enzymes, as you learn in chemistry, can be cyclases, kinases, phosphatases, and who knows what. So all those receptors that have some of these activities belong to this group. And finally, the third large group is those receptors that have ion channel activity, and this you have already discussed when you talked about ion channels. And the prominent example that you have discussed is the acetylcholine receptor, and of that the nicotinic or nicotinergic subtype. From the ion channel point of view, you call this ion channel a ligand-gated ion channel. Now I would like to turn the point of view around and call it a receptor which has an ion channel activity. But it, you have to understand, of course, is the very same thing. So what we will do now is today we are going to discuss the largest group of receptors that have no enzyme activity, and that's the group of receptors that are coupled to G proteins. And then in the next lecture we will discuss the second biggest group, which is nearly not as large as the G protein coupled receptor group, and those are the receptors that have tyrosine kinase activity, which is a subgroup of those with their own enzyme activity. And then after we've discussed these two blue categories in more detail, we will relate all the other categories to them later on, and at that point I will return to explanations and classifications, but there's no point going into it right now. The last thing that I wish to say here is that this categorization is somewhat different from what you find in the essential cell biology textbook, because essential cell biology does put the G protein coupled receptors into one big group, and everything else in these tables, like 1b, 1c, 2a, 2b, 2c, and 2d, is put into the other group, which number-wise is fairer, because this is such a huge group and very important, but logically it's a little flaky. So I would like you to keep this classification in mind, if possible, and not that in the Essential Cell Biology book. Okay, so let's go into detail about G protein coupled receptors. These receptors are also called 7 transmembrane receptors or 7 transmembrane domain receptors, because they have 7 alpha helices, numbered of course 1 through 7 very imaginatively, that go through the cell membrane, and there are loops inside the cell and outside the cell that connect them. They usually have their amino terminal on the extracellular space and their C-terminal or carboxy terminal on the inside. And because they start and go through the membrane and go back again and come and go and come and go, come and go, it's like a snake, so they also called serpentine receptors. The example through which I would like to introduce these receptors to you is a receptor which is particularly important when you have a stress reaction, a fight or flight reaction, for example, in a difficult exam, when you're being asked, okay, so tell me about G protein coupled receptors, and I say, oh, what should I say about G protein coupled receptors? So you get kind of a bit of stressful, and maybe if the question is not so easy as GPCR or G protein coupled receptors, but something more complicated or more esoteric, then your adrenal gland tends to squirt a handful of epinephrine or adrenaline molecules into your bloodstream, and we will follow now through how this epinephrine works in your various organs. But before we do that, let's just think a little bit about it. What do you need when you ask a difficult question in an exam? Of course, you need to think. You need to think hard, and for that, what you primarily need is glucose to your brain, so that you can actually think, because your brain does not eat anything else, just beautiful, nice, pure glucose. So we will look at the example of the liver, which is through its glycogen storage the major source of glucose that can be released into your bloodstream and used to feed your brain. In the liver, epinephrine binds to its receptor. Uh, it's called the beta adrenergic receptor. As you realize, I intercalatedly say either epinephrine or adrenaline. One is the Greek and the other is the Latin name for the same hormone and the same gland. So so epinephros means above the kidneys in Greek, and adrenos means to or next to the kidneys in Latin. Ad is next to or to, and dren, from which the English word renal comes from, is the kidney. So epinephrine or adrenaline are both used depending on British or US culture or who knows what. So please get used to both names. The receptor is adrenergic after adrenaline. The receptor binds this ligand, and it doesn't really do much. It only changes conformation, and then since the intercellular parts changes conformation, it can interact with other molecules and forward the signal, forward the message saying, okay, I have bound the epinephrine, so the signal has come in. Who does it forward the message to? Well, you already have learned about G proteins when you learned about nuclear transport. You learned about the RAN uh, proteins, and also we have mentioned during our studies on the cytoskeleton various proteins like RAC and Rho, which are also G proteins. G proteins are famous for binding GDP, guanosine diphosphate, and then when activated, throwing it away and exchanging it for GTP. The protein that we talk here about is also a G protein, but it's not monomeric like RAN or REC was, but it is a trimeric G protein, which is made of alpha, beta, and gamma subunits. As you realize, cell biologists have a great vivid imagination, so they name things like alpha, beta, gamma, 1, 2, 3, or maybe Roman 1, Roman 2, and Roman 3, or if they really get carried away, they may even call something A, B, and C, which is made of 3 different parts. So in this case, it's alpha, beta, and gamma. And the alpha subunit is the one that binds GDP. And when it interacts with the ligand-bound receptor, it releases GDP. Once it releases GDP, it will bind GTP simply because there's so much more GTP in the cytosol. So when GTP is bound, then this alpha subunit gets very happy. It's like you have a family with a daddy, a mommy, and a kid, and then daddy goes, buys a lottery, and hits the jackpot. So he's getting very happy, and out he goes into the night to have some fun, like this alpha subunit does go alone now, separated from the beta gamma, and it is meant to activate some effector molecule. In our case, in the liver, in the example of the beta adrenergic receptor, the example is this molecule, this blue molecule, which is called adenylate cyclase. So this adenylate cyclase earns its money by taking ATP and creating cyclic AMP from it, using the energy after cleavage of the inorganic pyrophosphate from ATP and closing the phosphate into a ring. That's why it becomes cyclic. So that's why it's called adenylate cyclase. Adenylate cyclase gets active by the GTP bound alpha subunit of the trimeric G protein. And also we have to remark that because this is an activating G protein, then it has a little index here after G, which is a S, letter S, which stands for stimulatory. So it is a stimulatory G protein alpha subunit. And at this point we really have to touch down on something very important in the whole concept of cellular signaling and regulation, and that is the principle of needing to shut it off if we switched it on. Otherwise it goes on and on and on, and there's no way to reopen the case and stimulate the system again. So what does this mean in the case of the G proteins? Remember that G proteins have GTPase activity. So, for example, you recall that when the RAN GTP exited from the nucleus into the cytosol, then the protein RAN GAP, which is the GTPase activated protein of RAN, stimulated it to cleave GTP, cut off an inorganic phosphate from GTP, and convert the GTP into GDP. The same thing happens to this G stimulatory alpha, but it doesn't need a GTPase activator protein. It actually very easily does it after a short time by itself. So it has a GTPase enzyme activity. It's like a self-timer. It gets activated, binds GTP, and then after some time it cleaves GTP, becomes inactive, leaves adenylate cyclase or any other effector molecule that it may be working on alone, and rejoins the beta and gamma subunits. So it's like this guy has spent all the money he won on the lottery in the night, had all the fun it could have, and then after losing all the money he went back home and said, please let me in, I will behave. And then the family is reunited here. The other thing that happened in the meanwhile was that the moment that the ligand-bound receptor stimulated the GTP exchange on the trimeric G protein, the conformational change that the alpha subunit causes by leaving the beta gamma makes it easier for the receptor to leave this beta gamma subunit and to release the ligand. So by the time of the cycle, through various switch off and negative feedback mechanisms, you restore the starting system with all the inactive participants of the story. So now we will examine what the cyclic AMP created by adenylate cyclase does. Cyclic AMP is a regulatory molecule that activates protein kinase type A. Protein kinases, as you recall, are enzymes that take ATP and cleave phosphate off and put them on various amino acid residues of these proteins. And the protein kinase A belongs to the group of serine-threonine kinases, which means that these kinases are specific for the aliphatic OH groups of amino acid residues, which are found on serine and threonine amino acids. It's a very uh, nicely regulated molecule because it has two catalytic subunits. These would be two catalytic subunits here, and two regulatory subunits. These green ones are the regulatory, and then each regulatory subunit has two binding sites for cyclic AMP. And when cyclic AMP binds to the regulatory subunit, then it releases the catalytic subunit, and these catalytic subunits are free to go, are active in this case, and are able to phosphorylate serine-threonine residues each. So the situation is roughly similar to me walking in the streets and having two really mad pit bulls on a leash. So this me walking, and these are the pit bulls on the leash in my hands, and then my kid comes across me, his hands full of ice cones with ice cream, and offers me. Of course, I love ice cream, so I will drop the leashes and grab two ice cones, and then the two mad pit bulls run off and bite everybody. So in a similar manner, these catalytic subunits would be running off and phosphorylating everybody when the regulatory subunit gets its ice cream in the form of cyclic AMP. But also in the way as I consume the ice cream and then run after the pit bulls and try to gather up the leashes and get them under control, the cyclic AMP that keeps the regulatory subunit busy and happy is consumed. And again, here we have to sort of refer to this requirement of switching on and switching off in biological systems, because cyclic AMP cannot forever exist, otherwise the cell would remain in a stimulated state. So there's an enzyme called cyclic AMP phosphodiesterase, which cuts one of the ester bonds of cyclic AMP, and therefore it is converted into linear or normal AMP. But then, of course, it doesn't bind anymore to the regulatory subunits here. So the equilibrium, which is between cyclic AMP and the regulatory subunits and their cyclic AMP bound form, is shifting back towards cyclic AMP dissociating, because its amount is being decreased by the phosphodiesterase. And because of this, the regulatory subunits eventually get rid of all the cyclic AMP, and then they can bind again the catalytic subunits and keep them in check so that they don't go around and don't phosphorylate everybody. As a brief reminder, I want to mention that phosphorylation that we talk about in this case is a reversible process. So once a phosphate is put on a protein by a protein kinase, like this protein kinase A that we discussed in the previous slide, it also can be chopped off in hydrolysis. So the chopping off is done by protein phosphatases. Why is this important? It's important because this process then is reversible and can be dynamically regulated based on the balance of kinase and phosphatase activity. And also it's important because it does change the conformation of proteins in a very profound way, even though there's only a very small piece of something put on them compared to the size of the protein. And this is just because the charge and the polarity of this phosphate group is so huge that it is able to alter the conformation of the protein very heavily. And of course that would be usually the tertiary structure of the protein that is most changed in these cases. So after this little detour of revision, let's see what's next. How does the whole sequence of producing sugar for your brains when you asked about the beta adrenergic receptor in an exam proceed? So we start by the adrenal gland releasing epinephrine. This goes around in the bloodstream and binds to its beta adrenergic receptor in the liver. This beta adrenergic receptor activates a stimulatory G protein, which activates in turn an adenylate cyclase. And the adenylate cyclase can create several cyclic AMP molecules each. So already here we see some amplification because a hormone-bound receptor can activate maybe a few adenylate cyclase molecules during its lifetime, but each of these few adenylate cyclases will create a lot of cyclic AMP, and each pair of cyclic AMP molecules will release an active catalytic subunit of protein kinase A, which is this yellow thingy-bingy, and each of these catalytic subunits can phosphorylate several other targets. In this particular case, in our example, the enzyme that's being phosphorylated is the so-called phosphorylase kinase, or in its complete name, it's glycogen phosphorylase kinase. This is a very ubiquitous enzyme which you will learn a lot more about in biochemistry, I assume. So at this point I only want to say that that it's an enzyme of 4 different subunits: alpha, beta, gamma, and delta. And one of these subunits, delta, is the K-modulin that you have learned with Professor Pani about when you learned about calcium-regulated processes. So this will tell you that in a very curious way, calcium itself could activate this glycogen phosphorylase kinase, but also the phosphorylation of 2 of its regulatory subunits by protein kinase A will also increase its kinase activity. And just in case for those of you interested, the structure is not as simple as 4 different subunits, but these 4 subunits gather up into a assembly of 4 of these subunits. So eventually it will be 16 different proteins joining up into a quaternary structure. So so it's a rather large enzyme. But in terms of our studies in cell biology, what we want to remember of all this is that, first of all, it's an enzyme with various subunits which has kinase activity. It is also a serine threonine kinase, and it can be activated by phosphorylation by protein kinase A, and also it can be activated by calcium. Now, what does this enzyme glycogen phosphorylase kinase do when it's activated? Its active form phosphorylates another kinase, which is glycogen phosphorylase. So glycogen, it's called glycogen phosphorylase kinase, is because it phosphorylates glycogen phosphorylase. And glycogen phosphorylase is an enzyme that breaks down glycogen into glucose 1 phosphate. So, so in the liver, if a single epinephrine binds to its receptor, it has the potential of generating several tens or even more of active glycogen phosphorylase kinases, which will create a whole bunch of glucose molecules that can be released into the bloodstream and feed your brain. So this kind of amplification is what is referred often to under the name of a cascade reaction, because one step causes the occurrence of the next step and the next step and the next step and so on and so forth. Just as in the waterfall, which is a cascade, the drops of water follow each other. So this was the description of one particular example, and particularly important example of how a G protein coupled receptor works. But as always, the situation is not as simple as one single stream of events, but even when we consider one single effector molecule in various cells, which is adenylate cyclase, which makes cyclic AMP, its regulation is far more complex than being regulated through a stimulatory G protein, through a beta adrenergic receptor, by epinephrine. Why do I say this? First of all, there are several other receptors, like the receptor for glucagon and also for adrenocorticotrophic hormone, ACTH, that bind to GPCR or G protein coupled receptors that activate stimulatory G proteins that can activate adenylate cyclase. This also tells you that glucagon, which is also a hyperglycemic hormone, just as epinephrine, can work through the same pathway. It can also stimulate the stimulatory G protein in the liver, which could stimulate adenylate cyclase, and then cyclic AMP activating protein kinase A will stimulate this glucose production cascade that we discussed a minute ago. I'm sorry I cannot go back with the slide, although in a live lecture I would, because the moment I roll the slide back, it erases the words that I've been speaking before. So just think back or roll it back once you are in the movie. Okay, so this is the input side on the stimulatory side of adenylate cyclase. But as we said, it's a major principle that things must be switched on and then should be switched off. There's also another major principle, which is that if possible, systems should be balanced like a seesaw. So in this case, adenylate cyclase is balanced not only by the GS alpha switching itself off by cleaving GTP and getting back to inactive GDP bound state, but also by having inhibitory G proteins, also trimeric G proteins, that can inhibit its function. And these inhibitory G proteins can also provide input through various receptors, and examples would be receptors for adenosine, opioids, cannabinoids, 5-hydroxytryptamine, and very interestingly also epinephrine. Now I highlighted this red, and especially these three things I highlighted red because I didn't give all these examples for the purpose that you actually do remember these for the exam. although later on you will need to remember them during your medical studies. But for now, for this course, I would only like you to remember epinephrine and glucagon. And this because these two work through the same pathway in the liver to generate glucose and let it go into your blood. So it's an example of studying and learning one pathway that we did in the previous slide or slides, and being able to apply it to yet another one and even more. So you learn one scheme, and then you put into that screen into that um, stream of events something else, and you have also a valid procedure. The other thing that I would like you to remember is that epinephrine can have different receptors. The beta adrenergic type will feed through stimulatory G proteins. The alpha adrenergic type will feed through inhibitory G proteins. Obviously, uh, in the same cell, it wouldn't make any sense to express both of these receptors, so both beta and alpha adrenergic, in the liver, because then the liver cell wouldn't know what to do. Should it activate adenylyl cyclase or should it inhibit adenylyl cyclase? But it makes sense to express different receptors for the same ligand in different parts of the body. How does that work? When you get into a stress situation, into a fight or flight situation, and your body is flooded with epinephrine, it is not only your liver that needs to produce sugar for your brain. Also, if that danger situation is not an exam but it's a physical danger, you might want to be running away, and then glucose is also needed by your muscles. But what you maybe also some of you have experienced is when you are in a few days stress preparing for an exam, and I don't really want to mention here your biophysics exam last semester, but about 60% of the people, when they are stressed for exams, they cannot really eat well. They they kind of don't have appetite, and food doesn't really wish to pass through them. And that's because they already have a high or reasonably higher than normal epinephrine level in the blood, and that through alpha adrenergic receptors uh, does several things that is responsible for these effects. For example, it slows down the passage of your intestines. Uh, in fact, it also, under a heavy stress situation, contracts some blood vessels which are in your extremities. So when you are in a stress situation, you don't need to be cozy and warm and have a soft and warm skin and be relaxed. Actually, you don't want any of your blood in your skin, because you want all your blood in your muscles so that you can run as fast as you can, and the rest of the blood you want in your lungs and your brain. So then what you say in this situation is that your periphery closes down. Periphery meaning the acral parts, your extremities like your ears, the skin on your fingers, and so on and so forth. Your nose gets very cold and stuff like this. So when you have a stress situation, you release a single hormone, epinephrine, and it seems to take care of everything. There's sugar for your brain and your muscles. There's also a dilation of your bronchi so that you can ventilate your lungs very well and supply yourself with oxygen. and they all feed into a fast escape. And at the same time, you also close down what you don't need, so you don't at this point need to eat and drink, because you don't need the extra food in your tummy when you want to run away from a lion, for example. And you also close down all the circulation in the parts of the body that don't need it momentarily. So then this system of multiplex receptors with multiplex G proteins can contribute to a single hormone causing the desired but seemingly opposite but together coherent effects throughout your body. Other things that what you want to mention that is that there are, of course, various adenylyl cyclases. Their regulation can be different, so there are subtypes, and and their regulation between different G protein alpha, beta, and gamma subunits can be different. But what you want to remember from this is that of G proteins there's various alpha subunits in cells. There are stimulatories, inhibitories, and others, and there are also several beta and gamma subtypes. And the cellular composition and also the types of effectors in the cell will determine what the overall output is. And of course, primarily the input, whether it's one receptor or another, feeding into the interpreting systems, is what is going to determine the direction of the cellular response. There are a few more issues that we want to mention on this complicated figure. One is that so far we've been treating the system as a ligand binding to a receptor which meets a G protein which activates an adenylyl cyclase. In a real life cell, of course, it is not so. There are many receptors in the membrane. There are also many membrane bound G proteins. There are also many membrane bound effectors. Also there are other effectors which are not membrane bound, different types. And they randomly diffuse around, or maybe not so randomly, because if you think about the membrane structure lectures and what you learned about lipid rafts in biophysics, you remember that there are some areas, specific areas of the membrane that have different compositions and used to segregate such receptors so that they can efficiently meet both their ligands and their effectors and their intermediary G proteins in their microenvironment. But what I'm wanting to emphasize especially is that because there's an abundance of all these molecules, there are many of them, they randomly diffuse, they randomly meet, and so it's possible that a receptor that does not bind ligand meets a G protein and then they collide but nothing happens. And in another case, a ligand bound receptor collides with a G protein and then it will very likely activate and cause the separation of the alpha subunit and the activation of it. So there's a lot of movement and random collision and meetings between proteins going on. And when you change the circumstances, like you add the ligand, then what you do is you simply shift the equilibrium of these processes towards other species. So there's always going to be some alpha subunits in the membrane that are GTP bound, but the vast majority without the ligand will be GDP bound and inactive. And conversely, when the ligand binds to the receptor, then the vast majority of alpha subunits will be converted to GDP bound active forms. So whenever we talk about singularities like the ligand binds to the receptor that activates the G protein, which activates the adenylyl cyclase, it describes the majority of events, the most abundant, the most likely events of which the most occur, because this majority will determine the fate of the cell during the next steps or next moments of the cell's life. And therefore, when you are asked a question in a test like GS alpha is in a GDP bound inactive form when there's no epinephrine binding to the beta adrenergic receptor, please don't say no, thinking that there will be out of 1 mole of GF alpha 16 molecules that are randomly GDP bound, but say yes, it is GDP bound and inactive because this is the majority, this is the central tendency, and this is what determines the cell's fate. And once more I would like to say that please think about this seesaw, this pendulum type balancing, that many systems which have a central character like this adenylyl cyclase will be balanced not only in auto shutting off or switch on switch off single inputs, but also by a balanced input of inhibitory and activatory molecules. So now once we have discussed this, a general scheme for adenylyl cyclase regulation and balancing, let us go back again to what happens after the fight or flight situation is over. And for that we will take now the example of the heart. We have so far generated glucose in the liver using epinephrine binding to the beta adrenergic receptor in the liver. But this sugar must actually get to your brain and your muscles, so you need to enhance your circulation as well. And that's through regulating the heart using the same epinephrine that we have already described to have many various effects on your body. In the heart, you also have a beta adrenergic receptor which binds epinephrine, which is coupled to the stimulatory alpha subunit of the trimeric G protein with some beta gamma subunits forming a trimer, and activation of the alpha causes the activation of adenylyl cyclase, which produces cyclic AMP, which activates protein kinase A, as we discussed. The protein kinase A has various targets in the heart as well, and we only mention one target, which is the plasma membrane calcium channel of the heart, which causes influx of sodium and calcium and depolarization and contraction. And phosphorylation by the protein kinase A, when you are in a fight or flight reaction, is going to enhance the operation of this channel in a way that it first of all lowers the activation threshold, so it will open at a lower depolarization voltage, and also the conductance will increase. So these two both lead to a stronger and faster heart rhythm and enhanced contraction and speeded up enhanced circulation. There are several other molecules that take part in this regulation, and it tends to be rather more complex than this simplified scheme, but this you will study in depth in physiology. So at this point, on the principle of less is more, we don't say more about this. What happens when the stress situation is over and you actually survived? Then it's time to go back to normal. And you have two different mechanisms for that. One is the spontaneous decay of this beta adrenergic stimulus, because the cyclic AMP will be degraded by cyclic AMP phosphodiesterase, as we discussed. So pKa catalytic subunits go back to their regularity, and then they won't phosphorylate channels anymore, and then phosphatases, which are always around and keep chopping off the phosphates, will turn back the regulation of this channel to normal. But that takes some time. However, as you recall from our high school studies, there's also another side of this nervous system, part of which is called the sympathetic nervous system, which contains the stimulatory epinephrine, norepinephrine release, and that is the parasympathetic nervous system, which balances it out and takes major part in the relaxed state of the body, where it recuperates and regains energies. And the major signal transducer or messenger molecule of the parasympathetic nervous system is acetylcholine. You discussed acetylcholine has two different types, as you studied it when you studied the ion channels. There's the nicotinic acetylcholine receptor, which contributes to muscle contraction, and there's also the so-called muscarinic or muscarinergic receptor, which is G protein coupled. This is a receptor that you would have called in the nervous system a metabotropic receptor, whereas the nicotinic receptor for acetylcholine in the neuromuscular junction, as you recall, would be called a ionotropic receptor, because that is an ion channel which is ligand gated from the point of view of ion channels, or it's a receptor with ion channel activity, receptor for acetylcholine. Now in the heart, there's this G protein coupled muscarinergic or muscarinic acetylcholine receptor, which couples to an inhibitory G protein. And the inhibitory G protein does two things here when it's time to relax. First of all, the alpha inhibitory inhibits the adenylyl cyclase, so that it counterbalances the still existing alpha stimulatory effect on the cyclase, and therefore inhibits this phosphorylation pathway. And the beta gamma subunits regulate an ion channel. This is a potassium channel which is opened by the beta gamma subunits, and opening the potassium channel leads to hyperpolarization of the cell, because potassium conductance increases and the membrane potential shifts towards the potassium equilibrium, which is about -90. So the heart muscle cell will hyperpolarize, And this hyperpolarization will make it more difficult for this channel to get activated and to initiate contraction. So hyperpolarization works against the heart cycle and decreases the speed and also decreases the strength of the contraction, and this is how you go back to normal heart rates and a normal rate of circulation. It is now time to step forward and and admit that adenylate cyclase, unfortunately, is not the only effector that is regulated by G proteins. There are many others, and adenylate cyclase was one example through which it is usually easy to understand how these receptors work. And we discussed the effects of epinephrine or adrenaline and glucagon on glycogenolysis in the liver, that means breakdown of glycogen and producing of glucose 1 phosphate and releasing sugar into the blood. Uh, these receptors can also lead to breakdown of fatty acids. Also, G stimulatory proteins can stimulate estrogen and progesterone synthesis in the female genitalia, and and then one last example for stimulatory G proteins would be the regulation of water reabsorption in the kidneys, which is regulated or enhanced by the antidiuretic hormone, or in other name, vasopressin. This you will learn in physiology in more detail. I only want to hint, but will never dare to ask in a test, that the stimulatory G protein in this case, when it's activated, also activates protein kinase A, and that phosphorylates a water channel, and aquaporin number 2, which is then placed in the membrane and is responsible for reabsorbing the primary filtrate in the nephron. But of course there are many words in this that you may not even have heard of, so I just refer to physiology, and then when you study physiology, maybe you can take this lecture and check this part out again if you need. And in also olfaction, of course, stimulatory G proteins play a central role. That must be mentioned. Now let's jump to other effectors, and one group of effectors are phospholipases, that are enzymes that cleave phospholipids, and these are activated by other G proteins, GO and GQ subtypes, rather than S in the case of adenylyl cyclase. So the beta type phospholipase C is activated by these, and we will discuss more details about phospholipase signaling in the next lecture. The third example of an enzyme which is regulated by G proteins is cyclic GMP phosphodiesterase, and that's regulated by the enzyme, by the G protein GT. T stands for transducin, and once you hear the word transducin, you remember, oh yes, of course, we learned this in biophysics. So you would need to revise your biophysics knowledge of how vision and the activity of rods and cones is regulated through the transducing protein, but I'm going to summarize it in the next slide for revision. And finally, the large group of ion channels that are mostly in the nervous system, the central nervous system especially, uh, are regulated by G proteins. So this would be the so-called metabotropic regulation in the nervous system, where ion channels are not directly ligand gated, but are regulated through various G proteins that are linked to various receptors, and these receptors would be for 5-hydroxytryptamine, gamma aminobutyric acid. Of course, there could be adrenergic receptors, especially alpha adrenergic receptors, but also beta, dopaminergic receptors, and the muscarinic receptors that we already discussed in the heart are also available in the central nervous system. And that basically gives you also a clue that if you don't know the answer to a question, then what will you answer? The answer you should always risk if you don't know anything at all is ion channels. Try to say ion channels. 50% of the case it works. So give an example of G protein regulated proteins. You say ion channels, cool. Give an example of a substrate for a protein kinase. You don't remember glycogen phosphorylase kinase. Doesn't matter. You say ion channels, and then it's going to be cool for the ion channel in the heart, and so on and so forth. But as joke aside, of course, ion channels are everywhere and very important, so that's kind of trivial that they will be involved in many different cellular processes. So on the next slide, then I'm going to explain briefly vision. So here is your revision of vision. As you recall, you learned about a protein called transducin, which really is a trimeric G protein made of alpha, beta, and gamma subunits, the alpha binding GDP. And if light hits the rod or the cone, then the protein called rhodopsin changes because its prosthetic group, which is 11-cis-retinal, will change into all transretinal, dissociate from the upper protein called opsin, together with retinal is called rhodopsin, and the freed opsin has a different structure, different conformation than in rhodopsin, so it is going to be activating this transducin protein, and transducin has an effector called cGMP phosphodiesterase, which was inactive before, but the GTP bound alpha subunit of transducin activates this effector, and then cGMP phosphodiesterase cleaves cyclic GMP into guanosine monophosphate, the linear guanosine monophosphate. And as you recall, cGMP kept sodium channels open and kept these rods and cones depolarized, so when cGMP is cleaved, then these sodium channels will close, and therefore rods and cones repolarize or hyperpolarize compared to their regular potential. So the whole process, of course, because you're supposed to be seeing through this, is triggered by the photon, So, so you're not surprised then to realize that this protein opsin is in fact a 7 transmembrane receptor, or a G protein coupled receptor, or a serpentine receptor, but it doesn't bind a real ligand. Rather, its ligand is a photon. So the conclusion is that we have a very funny system which is triggered by a photon, causes the conformational change of the receptor, which is opsin, but then opsin is coupled to a trimeric G protein transducin in the regular way that we learned during this lecture, where the alpha subunit exchanges GDP for GTP, activates an effector, and the effector does something, generates or degrades a second messenger, which then causes various effects, and thereby leads to a visual signal towards the brain. At this point, it is prudent to remind you of the important principles of signaling that we have discussed so far. So, first of all, we've seen a lot of normal processes that happen in cell communication and signaling, and one important message is that cannot be emphasized enough is that all the pathological conditions that you will be meeting during your career are based on the very same molecules and phenomena that we discuss here, except that this normal regulation that we discussed will be altered, and you will have to interfere to restore it or alter something else which balances out the change, which, as we discussed in the very first lecture on cell biology during the beginning of the semester, where we still were happily living our lives without various viruses, is that when you interfere on another point, rather than simply restore the very problem or restore the process to normal, but interfere somewhere else that balances out the previous change, then you are going to change so many other things that you always have to count on side effects, and you always have to put on a balance the good and the bad that you cause with a given treatment. The next thing that I took some time already to explain is that there's a lot of molecules interacting, and there are several outcomes possible with different probabilities. So in these lectures and in these figures on single molecule doing something to another single molecule schemes, we always convey the dominant or average outcome, and this is the one that when it's asked yes or no, it should be marked yes in multiple choice or a true and false test. Then there was the principle of if you switch something on, you must switch it off. Options are using internal timers like the G protein hydrolyzing the GTP bound to it, or you can have other molecules that shut it off. For example, the protein kinase A activation, which is caused by cyclic AMP, is shut off by another molecule, which is cyclic AMP phosphodiesterase, which chops off cyclic AMP, thereby stopping protein kinase A activation. There are many ways of switching things off, and one prominent mode would be negative feedback, and we will be learning a lot of negative feedback in the next lecture. I would just like to forward refer to this. And finally, uh, I also mentioned the seesaw principle, that regulation of central effector molecules is best done in a way that they both have activatory and inhibitory inputs. So now that we have discussed G proteins in the healthy situation and gave a lot of examples, I would like to show you two examples of alterations which happen in pathological conditions. Let me first of all remind you of the G protein cycle, where the trimeric G protein is assembled and the alpha subunit binds guanosine diphosphate GDP, and upon some particular stimulus, GDP would be released. So then the molecule similar to it, which is in excess in the cytosol, which is GDP, will be statistically a lot more likely to bind to it, and binding GTP changes the conformation of this alpha subunit, which then allows it to regulate various effectors. For example, we discussed adenine cyclase, and eventually there's an auto switch off mechanism where GTP is hydrolyzed, a phosphate is cleaved off, and the trimeric G protein returns to inactive trimeric form with the alpha subunit binding GDP. So one of the situations when this kind of regulation is altered is in the case of a disease called cholera. Cholera is caused by a bacterium called Vibrio cholerae, and this bacterium produces a toxin which belongs to the AB5 family of toxins, which, as the name suggests, would mean that it has a subunit, which usually is what is toxic to the body or to cells, and there's a B5 pentameric subunit which is responsible for binding. And where this B5 subunit binds is on the membrane of the microvilli of the small intestine, shown here on this image. And in the membrane there are clusters of GM1 gangliosides, which you see on this fluorescent picture here. These green spots are the GM1 ganglioside clusters, and once the toxin of the bacterium binds to them, the whole toxin will get internalized, and then inside the cell the toxin A subunit is released and does a nasty work. And the result of this nasty work is that people have a very severe diarrhea and losing their body fluids through their intestine, and this whole disease is centered on the gastrointestinal tract, of course, because this Vibrio cholerae is most feeling most home in the intestines, and that's where it secretes its cholera toxin. So what is the mechanism of the diarrhea that is caused by the Vibrio cholerae? You can observe on this next figure. Basically, the cholera toxin has a so-called ADP ribosylase activity, which means that it takes an NAD plus, cleaves it, and it's cleaving it in an asymmetric manner. So a nicotinamide is cleaved, and then from the NAD and ADP and a ribose is bound to it, is taken, which is the remaining part of the NAD in addition to nicotinamide, and this is covalently bound to the alpha subunit of the stimulatory G protein. What it causes is that it prevents the hydrolysis of GTP by the alpha subunit. So then this alpha subunit of the stimulatory G protein is permanently active. So it produces through adenylate cyclase a lot of cyclic AMP, and that activates protein kinase A. And protein kinase A phosphorylates various molecules, including the CFTR chloride channel, which you learned with ABC transporters, and also aquaporin proteins. So it will cause a chloride efflux from the intestinal cells into the lumen of the intestine, and through aquaporin water channels, water will passively follow, and thereby the body loses salty water, which causes diarrhea, and it must be treated. Of course, it's easy to treat it using antibiotics against the bacterium, but in regions where this is not available, the so-called oral rehydration therapy can be utilized, where they have salty water drunken by the patients for those 1 or 2 weeks, during which the immune system learns to fight the bacterium, and it's the cheapest and not very convenient but certainly efficient way of overcoming the infection without fatalities. I also would like to show you a very similar disease, which is the whooping cough, or pertussis in the Latin name, and it's called by the bacterium Bordetella pertussis. It's a very similar mechanism that works here. So pertussis toxin is also an AB5 structure, and S1, which is the enzyme which has ADP ribosylase activity, as does the cholera toxin A subunit, and the pentameric S2, S6, which dock on the surface of cells and then get internalized so that the toxin can work from the inside. The difference between cholera and this is that the pertussis bacterium starts inside the body on the respiratory tract, and also the toxin can be found also in the blood circulation, so it has multiple effects on the body. The ADP ribosylation that the pertussis toxin does concerns also G protein, but there's a very big difference, namely that this G protein is an inhibitory G protein. However, also the ADP ribosylation happens in the inactive GDP bound state, so there's no way to progress to active inhibitory form, exchanging the GDP to GTP. So the cycle cannot progress between active and inactive, but the GDP bound inactive is fixed. And that also will cause the production of excessive amounts of cyclic AMP and the activation of protein kinase A. So overall, depending on which organ or organ system is concerned, you will have a lot of negative effects like immunosuppression, inhibition of phagocytosis, the increased permeability of blood vessels, which causes the release of fluids into the extracellular space, into the interstitium, decrease of blood pressure, which can result in hypovolemic shock, which you will learn later in your medical studies. It also, by affecting the cyclic AMP levels, can cause hypoglycemia and coagulation by activating platelets, which can lead to thrombosis throughout the circulation. Usually it's called a disseminated intravascular coagulopathy, or DIC, which can be very easily lethal as well. So it has a multi-system effect, can cause multi-organ failure, and it has the same mechanism. What I as cholera toxin, except it influences many different parts of the body. The the comparison in terms of molecular mechanism is that in this case, ADP ribosylation by the toxin causes the inhibition of an inhibitory molecule. So in terms of a car, it's like you cut the brakes. And if you cut the brakes and you put the car on a slope, then it's going to roll down. So this is opposed to cholera, where you actually keep the activating G protein in the active state, which is more like gluing a brick on the gas pedal, so you keep the car running by that. But whichever the mechanism, the car is going to be running. In this case, this running car is adenylyl cyclase, which is going to produce a lot of cyclic AMP, activate protein kinase A, and then yield all the various physiological effects of protein kinase A activation, depending on the organ or organ system that is concerned. So thank you very much, and I'm coming back with the next lecture soon.